Okay. Ladies and gentlemen, a very warm welcome to all of you. It's really a pleasure to have all of you here today. And I know there are a number of the participants that are joining us online via Zoom. And we had some fantastic weather in Tartu in the last 2-3 days. Even at the moment, it is drizzling a bit. But the drizzle with the combination of the yellow, red, orange leaves, it makes it even how can I say, Tartu is romantic, but is even making it more romantic, putting it in a very special space for us to start really speaking about One Health and how we have tried to bring impact, innovation, and the path forward. And definitely in doing this job, we are going to have a great program that has been actually prepared for you to have it. And I'm not going to go through the program, all the things one by one, but I want to say that for reaching to these aims and having this program, we have a great leader too. Professor Ulle Jakma, our Rector Magnificus, that is going to open the meeting and we would be able to follow her lead. Please, Ulle.
Dear partners, colleagues and friends, it is my great pleasure and honor to welcome you to the final conference of the One Health Boost project. Today, we celebrate the achievements of a remarkable journey, and this is a journey of collaboration, growth, and shared ambitions in the field of One Health. One Health is one of the 4 strategic priority areas in the Estonian University of Life Sciences research agenda. Our ambition is to become a prominent center of excellence in One Health research, both within the European Union and globally. Veterinary medicine is one of our flagship disciplines where we have critical mass, expertise, and experience to lead. But One Health is not only about veterinary medicine. To truly excel in this field, we must embrace inter- and multidisciplinary collaboration that connects human and animal health, biosecurity, and the resilience of our ecosystems. One Health Boost was launched with a clear and ambitious goal to strengthen the research, innovation, and management capacities of the Estonian University of Life Sciences. And together with our esteemed partners, the University of Tartu, the University of Helsinki, and the University of Milan, we have worked in close and effective collaboration to build a strong foundation for the excellence in One Health. Now, almost 3 years later, we can proudly say that our joint efforts and focused approach have significantly strengthened Estonia's expertise and visibility in One Health. We have deepened our integration into international networks, promoted One Health careers, and enhanced our institutional attractiveness. Our ability to lead proposals and attract funding has grown, and we have taken concrete steps to translate research into real-world innovation, building industry links and exploring business models. And our public communication and policy dialogue have helped raise awareness of One Health and its importance to society. And I would say that One Health is not just a modern phrase, that may fade over time. It is a principle that reflects countless connections that no single project or even single institution can fully address alone. But it is a principle we can apply together to shape a healthier and more resilient future. And most important, the journey does not end here. The next strategic steps will ensure that the impact of this project continues well beyond its formal conclusion. And now I would like to express my heartfelt thanks to all our partners, researchers, students, and all project team members. Your dedication and spirit of collaboration have been the driving force behind the Onehealth Boost success. Biggest thanks to Professor Alireza Fazeli, who has been a dedicated and experienced leader and a promoter of Onehealth initiative in Estonia. I wish you all a meaningful and inspiring conference day, full of ideas, and I am sure that you will take Onehealth research to the next level. Thank you.
Thank you very much, Ole. I have the pleasure also to work in the Institute of Veterinary Medicine and Animal Sciences. And this institute is headed and directed by Professor Thomas Thiarts. And Thomas has been really a great support of Onehealth. And in addition to that, he has been also a very good friend, especially at the times that you are completely low and you need somebody to take you up. Thomas, please.
Yeah, sorry, I didn't recognize myself over there, but I don't know from where you found this picture. Okay, but thank you for inviting me here. Dear colleagues, rector, also dear friend Alireza, I'm happy that on period, on project is almost at the end. And in spite of hectic times and in spite of a lot of things to do, this Onehealth lineage or just the focus has been accompanied us all over the recent years. So it means Onehealth approach is expected or anticipated wherever level we are imagine. This week it was talk about Onehealth approach in undergraduate studies. For example, recently meeting different ministries, ministry officers. On another way, we end with this Onehealth approach. Just setting up our new goals in near future and important tasks of different partnership. I'm most happy that we have here a lot of colleagues who supports such developments at our university. It's worth to mention that recently at our University Senate we already passed first reading or idea to establish Onehealth Center at our university to make it more noticeable, more visible outside and declare that we are here to supervise and lead the direction of Onehealth approach. So it makes me happy, and I'm also happy because of you, that just giving up, not giving up, but holding up high the Onehealth flagship. So thank you very much for your cooperation, and we look forward that there will be a lot of follow-up things. So, and a special thanks to our partners, Helsinki, Milan. Several times I had the opportunity to visit your places and discuss different developments. So all together we can do great things. So thank you.
Great. Thank you very much, Thomas, for your kind words. I think now the first part of the programs start, and I'm going to speak about the overview of the project Onehealth, that you know what we have been doing. And by that, I want to at first to introduce myself. My name is Ali Reza Fazili, and I am now something around 4 years that I am working in the Estonian University of Life Sciences, and started by working on the comparative medicine era chair. And then during the time of this comparative medicine era chair that I started to work in the molecule, we also, as one of the aims of the comparative medicine, that is a subsection of Onehealth, we try to apply for grants and to be able to somehow do the further research in Onehealth. And one of those grants that we somehow tried to apply and get was OH Boost, that is Onehealth Boost. The work on Onehealth Boost started probably something in 2021, and then 2022 was the height of the amount of the work that we put in it. And we were actually, by September 2022, we were successful. We knew that we have the success, and then we have started to do the thing. Then 2023, the project started. The project is actually, there are several partners in the project. The main leading partner of the project is Estonian University of Life Sciences, but at the same time, close to us, we have the Tartu University. And that is one of the strengths of this city, Tartu. We have two universities. One of the universities, Tartu University, that is oldest university, probably in, I can say maybe even in Europe, in one of the oldest ones with more than 400 years, and also having a very successful history of medicine. And then at the same time, we have Estonian University of Life Sciences with a very good success stories in the life sciences, including veterinary medicine. And it is nearly now 9 years I am in Estonia. Before being here, I have been in England. I have been working also in the Netherlands. I find it in the Netherlands, particularly in Utrecht, medical school was the other side of the street. But the amount of the interaction I see here between the medics and the vets, I didn't see it even in the Netherlands, that it is as much as that. Hence, I think partially, maybe it is a part of the Estonian culture that they speak, they work together, and they are open to at least discussion. That is, maybe if you speak about this with Estonians, they will say, no. During the COVID time, we were forced to be 2 meters apart from each other. We are usually 5 meters apart from each other. But that is not true. In my eyes, as somebody that comes from outside this country to this country, Estonians have the tradition of maybe not speaking, but working. If you go to them and try to do this, you are able to work. Hence, that we managed to do. The EMU and the Tartu, we were close and became closer during this project. At the same time, this project gave us the possibility to reach to Helsinki. Helsinki or Finland is, I have to say, it is a comfort zone for Estonians. Maybe because historically, you guys, you know each other. From the language point of view, your languages is also very close to each other. and all that. And also, if I say in other parts of Europe that I work in Estonia, everyone is looking a bit strange. Where is even Estonia and this and that? But for you guys from Helsinki or from Finland, Estonia is a non-entity. And that makes collaboration, cooperation with you simpler and easier. But I have to say, during the OH, we become even more closer to each other. And in the middle of this, we suddenly also started to outreach to other side of the Europe, south of Europe, near Mediterranean Sea. And this has been thanks to the contacts that I had with Tiziana. Me and Tiziana, we have been in 3 different coast projects. The first one, I was the chair, and Professor Gandolfi, also from University of Milan, was the co-chair. Then the second one, I became co-chair, and Tiziana was leading one of the work groups. And the third one, Tiziana was the leader, I was the co-chair. Then we had a very good connection with each other, and that was excellent to approach her and somehow bring the South Italy to ours. And hence it was, now looking back, it was a great combination of putting different geographical part, different cultural, different experiences together. And in all of this, the goals that we had was to actually improve and as much as possible somehow to increase the One Health area, to increase the research innovation excellence in One Health, and also improving the management administration capacities of Estonian University of Life Sciences in this field. And for doing this, we had generally 5 objectives. One of the objectives was regarding the scientific excellence. The other one was to be able to conduct exploratory research that would be in One Health, focusing on different areas that maybe we were not very strong in them before the OH boost. And of course, you cannot nowadays do research without considering the management of research. Probably you would see it also, there is a lot of bureaucracy, a lot of administration, a lot of issues involved in the research. And for that, you need to have that structure that supports researchers to be able to do their job. And that was one of the things we wanted to learn during this project. And you will see later when I speak about the different part of the project, we had that opportunity. Then the project was not only working with scientists, but also working with those people that are running the admin of the research and admin of how creating that environment. The fourth objective was about how can we have support of the young researchers? How can we make the life better for them? And how we can somehow create an environment that would make them to somehow nourished and be somehow enjoying what they do, and also at the same time working further with us. And finally, this is something that I have found it extremely important, especially in a society like Estonia. You need to also educate the whole community. And that education is not just only at the science space, but outreaching to the normal people and trying to somehow bring that out. And that was the number 5 objectives that we definitely needed to learn from it. I have to say that in each of these objectives, we had the support of the partners, that each of them were actually experts in and had excellence outcomes. And that was very, very useful for us to gain from them. But the project would not have been successful if we did not have an excellent team. People that came together, worked together, and this was whether local people working in Estonia or the people that the universities that somehow join us. I think nearly everyone that I see in this picture is today in this room, except Professor Gandolfi, that I really dearly miss him. But yeah, the problem is when you are a very good scientist and you are somehow having a lot of success, you are constantly going from one place to another place, it is hard to somehow have health. I hope Fulvio is now there, is hearing us, but we have the representative. But I really, really grateful to everyone that somehow worked and helped and participated in this project. I think all we agree it was a wonderful team. And I am nearly 100% sure after this project, we would continue. It somehow bounded us together, and it also created an understanding of each other. One of the things I have learned in my work is we are like the fingers of the hand. We are very necessary to work with each other or collaborate or so, but each of us is different, and that individuality is important, because that individuality would allow some of the things be done by some people better than the other people. Hence, it is always good to, as a leader, you have to learn who is doing the certain particular jobs better and promote them and to somehow help them to do that. But trying to make one size to fit everyone is not a very good idea. And that usually fails and doesn't work. And that individuality we try to also have in this project and try to use it in the structure of the project that we had. We had generally 6 working programs. One of them, the first one, was about the management, how to, in the working group, actually, the working group 6 was about the project management that was somehow covering everything. And then we had the first working group that was especially about the scientific excellence, boosting the Estonian University of Life Sciences scientific excellence. Then the second one was having a real example of doing research, that was in the second WP2, and then increasing EMU research and innovation capacity. At the same time, the fourth one was the supporting the next generation. And fifth one, that was actually cross going through all the other work packages, was about communication, dissemination, and exploitation. I'm just thinking about the thing. Then as we somehow did go through these packages and through these work packages, we had key results. We had the point that we have tried to bring in here. And for that, in the work package 1, what we managed to get was to gain competences that we didn't have before. For example, advanced training in metagenomic, lipidomics, and 3D cultures. These were the things that we gained from Helsinki and also University of Milan. At the same time, we have tried to upgrade our research developmental plans by taking examples of these institutions, what they have and what we can get from them, and also thinking about what can be the area of research in Onehealth for future that we can somehow progress and go to them. The same also in the work group and work package 2. I think it is a bit redundancy. If I speak about all these points here, you are going to hear it during this today in the afternoon about all the research activities. And so we have examples of it. Since I have a love for extracellular vesicles, it is, you find it that it is very much going towards extracellular vesicles. That's, I apologize for that. But at the same time, I think it is great. I was yesterday night, middle of the night, speaking with Romanian representatives of university there for a new application we are doing it. She was saying to me, yes, I'm reading about the extracellular vesicles and the things that you are doing in relation to Onehealth. And she was saying, it looks to me like science fiction, but I'm very, very happy to get involved and go further in it. Hence, that was a science fiction that we tried to bring it to the reality. After that, in the working group 3, we tried to strengthen the administrative and other supportive parts of the university. You would also hear about that, what we did and how we tried to. You have some examples that would come here and you speak. And of course, the core work that we tried to do, and a lot of work and the energy was put into it, was to develop and help with the next generation of the scientists that can somehow follow and can go further with the research and the Onehealth approach in the Estonian University of Life Sciences. And finally, we have the communication and dissemination. In this respect, we had Tiziana with her very, very long-standing experience in communicating science to ordinary people and different parts of the society. That was very, very useful. As a result of that, I think now we have become a companion of AHA, the science museum that we have in this city. And for quite a lot of their projects now, they are asking for our help and our somehow advice to get involved. And finally, yeah, project management. Project management is not simple, especially European projects like this, and to put everything together and reach to the meaningful ideas and so on. And that has been also one part that we have tried to excel and become better and better. Then if I want to summarize the main outcomes that we had from this project was gaining expertise and visibility, strengthening the Onehealth experience and research excellence at both Estonian University of Life Sciences and also University of Tartu, increasing integration of Onehealth to the networks and academic visibility, at the same time promoting Onehealth careers and improve institutional achievement and attractiveness of the Estonian University of Life Sciences. Regarding to the capacity and impact, we definitely boosted the ability to lead proposals and attract fundings. In some of the slides I would have, you will see the number of the applications that we did, and even many of you that are sitting here, you have been co-partners of us into these applications. Nowadays, the getting funding is not an easy job. We usually apply for the success rate has come down and down, but we apply, my own statistics is from every 9 application getting 1. But also at the same time, the application that we apply is not anymore a smaller application. We are now speaking about millions of them. Hence, it's, how can I say, it is, if one would be accepted, it somehow pays for all the time that we have put. But it is becoming a very, very hard business. And OH Boost was very helpful for us to find new ways, new niches that we can somehow apply and go together. And that was partially or mainly due to the fact that we teamed up together from Milan to Helsinki and also to the UT, and together we worked. Outreach and collaboration, as I said, I have already mentioned AHA, but we didn't only just do that. We did go to Arvamus Festival. That Arvamus Festival was an amazing experience for me to be able to get engaged and get it. Nowadays, thanks to Onehealth, we are even collaborating with CAPO. That is the internal security organization in Estonia. Never I thought as a veterinarian I would end up to collaborate, cooperate with internal security system, but they are actually more happy than me being with them, because we have expertise that they need us as that. They think that we are good for the future and also the health and wealth and also the security of the country. Then that is all have been as a result of outreach and collaboration and dissemination that we brought there. And then they approached us and we have started to collaborate, cooperate. And we are going through this path and we are somehow trying to improve ourselves. But if I want to give a bit of what has been the secret of the success and what has been the reasons that we have been successful in these last 3 years to work together, one of the main reasons has been the fact that we had clear goals and objectives. We knew what we somehow want to achieve and where to achieve it. At the same time, a strong teamwork, and that strong teamwork came through the very, very good communication. And at the same time, having stakeholders that really try to engage. And we partially, this is also going back to the fact that we were a bit known to each other. We were not unknown entities. We knew who we are, we knew of each other interests. And then as a result of that, we just boosted that active engagement. And finally, having a very smart outreach and visibility. And in that smart outreach and visibility, I have to bring the role of the younger members of the team. They did a fantastic job. And they also, still they are doing a fantastic job. Just the last Coffee Scientific that we had the open day of the researchers that was happening, I think, 2 weeks ago or 1 week ago. I was even myself not here. I was getting the videos and pictures of the members of the group that are interacting with the public. I was just wonderful. And this has been all the result of the work of the Onehealth Boost, that we created a team that is not only hands, but also brains. It works autonomously, individually, but knows what is the aim, what has to be done, and just goes forward. And of course, for me, in the middle of this, innovation, that is what we said, but at the same time, sustainable planning. I consider my job to be trying to find money and my team to spend the money. I think many people here would have the same somehow feeling as, but each of us should do what is good at and what can somehow do or what can go and get it. Then that has been probably these 7 points has been the secret of the success of the project, if I can say so. And in this regard, I have to say we definitely have had some very good example of internal impact that we have brought to the system, both for Estonian University of Life Sciences and Tartu University. Apart from the fact that now I don't feel that we are even two universities. We are really working so close together and so nicely, and we are complementing each other. We are not competing with each other. There are certain areas that UT is strong in. There are certain areas that we in EMU we are strong in, but we have learned to collaborate and cooperate with each other. And that has been a very, very nice outcome of this project, in my opinion. At the same time, we have been able to train researchers and PhD students to be involved in this. At the same time, also, our researchers and PhD students have become more open to the outside world, and particularly the other parts of Europe. All the visits that we had, all of that kind of thing, gave them the ability to taste another environment and be able also to see the other side. Is probably always the grass is greener in the other side, but it is not somehow perfect. Everything is there. They have to deal also with the problems that we have to deal with here, and all that to go together. And as I mentioned before, we have developed a very good project team that is very well working together. And in the middle of this, we had definitely, I think, had external impact. This picture, I think, by itself somehow says it is. We were in Brussels, and we were in the seat of the Estonian government in Brussels, and we see Ule, we see Irja Lutzer, we see Signe Ratso, that is the commissioner there, and myself there. We catapulted ourselves to the places that before this, as a relatively a small university in a very small city in Eastern Europe, we didn't thought that we can somehow reach to that parts and getting such audience listening and coming. And that has been partially thanks to the project and the work that we did here. As a result of that, this international research community engagement also with the stakeholders and definitely with the general public has been splendid, and it is still ongoing in our work. Finally, we have some numbers that we have put together for you. Some of them, Ursula said to me that some of these numbers are really to laugh or maybe to make a joke about it, but some of them are real ones. For example, in 36 months of the project duration, we had 6,000 participants in all different activities that they take part. We had 50 press releases. We had 10,000 people that online we have reached, whether through our websites or whether through our social media. 120 visits, trainings, meetings organized. Can you imagine in the last year? And how much work is there? 20 deliverables completed, 9 new membership of Onehealth networks we have achieved, 18 outreach events organized, 17 conferences, presentations, 20 scientific publications, 20 meetings with the stakeholders, 40 team photos taken during this. That is a very, very important measure you have to bring. At the same time, 100 urgent emails taken during the meetings, even maybe at this moment of the time that we are in this meeting, they are answering the urgent emails there. And 20 plus discussions about Onehealth translation to Estonia, and 100 times someone on Zoom said, "Can you hear me? Can you see my screen?" That is something familiar. But apart from, some of it maybe laughing about it, it has become our daily life. It has been a tremendous amount of work, a tremendous amount of job, and a tremendous amount of achievement. And we have not been able to reach here without the help, collaboration, cooperation that we had all together. And I have to say, we have been in the mighty hand of a project assistant that is sitting there with the red coat and has been excellent in sending constantly emails, everything, hammering it on our heads, or at least to my head. Ali Reza, you have to do this. Ali Reza, you have to do that. Hence, if ever you come into my position, the first thing that you do, you go to find a project assistant that can be open to you and can tell you what you're not doing wrong, and be receptive to what she's saying to you and listen to her. It would, at the end, it is better for your own situation. I see some people here already. But in any case, I don't want to take more of your time. I'm sorry, maybe I spoke more than what I have had to speak. Okay, I want to thank everyone, all of you. You don't know how happy I am and how grateful I am to all the works that you have done. And yeah, I am really, really privileged. This is the second time today I am saying that I'm privileged to work with a great team. Just today, we had another meeting finished at around 1 o'clock in Vanamune Theater. We had participants from 35 different countries and nearly 300 participants coming to Tartu to speak about extracellular vesicles. And I had my team coming on the stage, and I said to them that this is the best team, the dream team that you can imagine. I have it with the Onehealth and with the OH Boost. I am still another time in such a position. I have the best team that anyone can imagine. Thank you very much for your attention. We have Christy, one of our leader from the university.
Hello, nice to see you all here. And now we will continue our agenda with the research part. And as Alireza already said, that the research was concentrating and focusing mostly on extracellular vesicles. And on the agenda, you can see that today there will be presentations from all the partners, one from each partner, and they are related one or the other way with EVs. So we will start with the first presentation from Professor Thomas Orro, who has had his degree in veterinary medicine, and after that the PhD completed at the University of Helsinki. More than 10 years, he has been serving as a professor of clinical veterinary immunology in the Estonian University of Life Sciences, and his research is focusing mainly on inflammatory response and acute phase proteins in neonatal animals. And today he will present the results that are related also to systemic inflammatory response in lambs and calves, and of course with EVs. So.
Tere. I hope somebody is looking at the clock. I had a habit to everything goes wrong as you see. Okay. Luckily, I have new glasses I can see from here. Okay, systemic inflammation, something beginning. So this is the background of everything we are looking for. We are looking at the so-called acute phase response. Don't be bothered about the word acute. This is confusing sometimes, but basic principle is that whenever there is some kind of tissue damage, immunological processes, infections, trauma, etc., there will be a cascade of processes which will influence all the body. And different kind of processes will start. It will influence everything in the body. And the main thing, reason of this response is to get rid of and treat injuries, tissue damage. Okay? In a sense, it's non-specific. Okay? And which means this is a part of innate immune system and not so much of the adaptive one. Although innate immune system we need this before adaptive immune system can work. What we use a markers. We use acute phase protein as a marker of this general inflammatory response. And now we need to think about that acute phase proteins we are talking about now is systemic one, which means they are produced in liver. But this is not the only one. Acute phase proteins are produced also in local tissues. And but right now I will not going to talk about that because in a sense they are controversial with each other. So we talk about systemic response, all body response, host as a whole response in that sense. Now, there is another important part. So acute phase proteins, these are produced in liver, but the initiation of this production comes from the site of injury, and so-called pro-inflammatory cytokines has the major key effect to that. Okay. Now to the case. So this is 13 calves, first 3 weeks of their life. What I did, I measured serum amyloid, one of the acute phase proteins, concentrations. If you take the first slide, and you know that serum amyloid as acute phase proteins means that somewhere there is enough tissue damage. What do you think now if you look at this picture? What's come to your mind? If serum amyloid is a marker of tissue damage, what it means, what this picture means? So the damage decreases with age. It's very good for me because I will age all the time. Okay? Every other talks about that. What do you mean? Yes, it's highly fluctuating. And if you look at this, you may think that those 13 cows were very sick during the first 2-3 weeks. No, they were not sick at all. They were totally happy, nice dairy cows. Okay. And now the problem starts for me because I started my PhD to think that I can use this marker as a sign of problems. You cannot do that. We are not very sure yet what is the meaning of this, but this is universal. The same pattern happens with acute phase proteins in babies, piglets. Okay? We have shown this many times in lambs, in cows many times, and in reindeer. Okay? So, I have a theory. If you look at those changes, if you took them apart, what you can see is that many of these curves have two peaks. It goes down, then it goes up, it goes up, it goes down, and it goes down again. This is very frequent pattern with that. So you can think now that there is actually two different kind of processes lying with each other, on top of each other. There is one process which we can be is a physiological one. The idea is that it represents a preparedness to the attack. So cerebromyelin A is very important immunomodulator related to the innate immunity, etc., etc. So it makes sense that there is enough cerebromyelin A available when something happens. This is the main explanation to that thing, okay? However, it will not gonna exclude that behind of that really something happens and you will have the next change to that. So the basic problem as a clinical marker is that those two things are overlapped. And I was not even sure what it means until I start regularly from my collected databases look at the future performance. So for that, those cows, reindeer cows, lambs, we started to look the association of the level of those acute phase proteins with the weight gain development. And what we find out, that the critical period of 2 to 3 weeks, if the marker is higher, it's quite probable that the weight gain is lower. And this can extend until the slaughter of the animal or until the other things. So it's very long-lasting effect. Something happens, we don't know what, during the 2nd and 3rd week, and it has some impact, sometimes quite considerable, for the rest of the life of this animal. There are different theories. Maybe the strongest one, I think, is related to the immunomodulation at a very important and critical time when passive immunity is going away and your own immune response related to the active immunity of neonates will not start that very well yet. So this opportunistic and dangerous window is actually overlined at 2nd and 3rd week of the life. So we have shown the associations. We know across the animals and different data sets, there is always have been, most of the cases, in some cases like calves all the time, in lambs, in reindeer calves, even now I know in piglets, this can be a problem. The next question is mechanism. What is the mechanism? And we figured it out, but maybe using the EVs, we can actually look closer to the possible influences and mechanism which is behind that. So we used some data sets we have collected, and first I talk about lambs, after I talk about calves. So this was separated EVs from fecal samples from 17 days old lambs. And this we designed especially looking at the low and high systemic response, low serum already, and at age, and high serum already group. So the idea will be that can we look at the metagenomic results profile of those samples and connect them with the systemic inflammatory response we measure. Okay. And the other idea, if we do this with EVs, would we have more detailed information coming out of this? That was the idea of this study. And then we did the same thing, calves. With calves, although all the same things were exactly the same. Same SEI is influencing the weight gain similar way as lambs. We decided to concentrate to the Cryptosporidium infection and see if in the deep level we can differentiate between Cryptosporidium positive and Cryptosporidium negative calves. So we use this data. First results I want to show you is that I was, in retrospect, I'm actually happy what happened with lambs, because as you see here, regardless of the group, low or high inflammatory level, the metagenomic profiles of fecal samples and EVs were roughly the same. Okay, which tells me that at least EVs represent the microbiota of the sample. Okay. And then we did the initial analysis. So for that, we used this kind of causal models. So the causal models, there are different group of these, but the causal model will evaluate the stability of the quite extensive manipulation of the original data. Why they call this causal model? Because the highest stability is indication of causality. It will not tell you directly, but these models are developed for that. And how they usually use is that with very big dimensional data, whatever the data is, microbiota data in our case is the good example. So this is used to restrict those genera you are actually looking for to get more meaningful and more smaller data to put to your analysis. And usually these results are used for separate analysis types. It's regression models or PCA, etc. That's the meaning of those models. There are different groups of these models. They have very different techniques to use, but we used here this model, elastic network model with multiple permutation and jackknife procedure. Anyway, and now there are results. We had a weight gain. We had information about pro-inflammatory cytokines, in this case, interferon gamma, and we have ceramide A. So the higher stability, the more causal relationship can be anticipated. What we are looking for is the overlaps and the direction of association, and to look is this meaningful? And if you look at this is meaningful in the sense that if something is negatively associated, you can look this in coefficients with outcome, in that case, daily weight gain, then it means by theory, it must be positive association with cytokines and ceramide A. And this is the case here. The other thing, you want to see that the association is in the same direction in cytokine and ceramide A. Of course, more cytokine means more ECA. Very good. So there are different picture. If you look, this was EVs. If you look at the same thing in fecal samples, you will see the different, the same patterns. So the results goes together nicely, although this table tells you a different story. So in the end, I'm convinced, I cannot say it's true, I'm convinced that using EVs and fecal samples, we can actually get deeper. Is this good result? If you believe as I believe in, yes? Okay? To be honest, I need to finish here, and it's very good, because if we go to the calves, then everything is different. It's not working at all. I don't know why. I have some ideas, but because I am lack of the time, I luckily can skip this. You see, and show you this picture. So do you know what animal it is? Somebody, Finns can shut up, but others. Do you recognize this? Reindeer. Actually, the correct name in Lapland is Porovasa. Okay, reindeer calf. So this picture is taken over 20 years ago. And today, when I listen to Alireza, I realize that I have done, not knowingly, I have done this OHA boost program. From the beginning, I have done this 25 years ago. I started with this program in cooperation with Helsinki University. It's my life. So here is all started. This OHA boost project started there, at least for me.
You hear me? So, not, then we thank, and during the coffee break, you can ask all the questions from the Thomas get the answers. I'm sure. Thank you.
So, and it's time to move on to our next partner. Ratko Avi is coming from University of Tartu. He's Associate Professor of Medical Virology. His earlier research has been focusing on HIV and its co-infections, drug resistance and host genetics. However, current work is concentrating on human gut microbiome, particularly on virome and mobile elements. And his presentation today is also concentrating on virome, in particularly on the bacteriophages and extracellular vesicles.
Thank you. Yes, thanks. My presentation, as said, is about viruses, especially bacteriophages in extracellular vesicles. And we wanted to know how extracellular vesicles carry or bind different viruses, especially bacteriophages, because they are very important and interesting factors in shaping microbiome development in young children, because viruses inhabit at least human gastrointestinal tract at the same time as first bacterium, and most probably the proviruses are the most or the first places where viruses emerge into the gastrointestinal tract. So we wanted to know what is the role of extracellular vesicles in carrying these phages and to modulating the phages fraction, as well as metabolic and taxonomic composition of microbes in gastrointestinal tract. We had 4 infants, 4 weeks old, and we analyzed 2 types of samples, bulk fecal metagenomic DNA and also the extracellular vesicles fraction in the same samples. Extracellular vesicles enrichment was done by our good colleagues in the Estonian University of Life Sciences. They used size exclusion chromatography to separate these extracellular vesicles from fecal samples. They made some characterization also for them, and thereafter we sequenced these 2 fractions. We used NF-core MAG for read quality control, as well as assembly, and therefore we analyzed these assembled contigs to do bioinformatics software meant for viral detection, Genomad and VirSorter 2, and thereafter we analyze the quality of these viral sequences. And what did we get? The number of contigs varied a little bit. We got about 2,500 to more than 8,000 contigs, longer than 100 base pair. but the distribution of different length of contigs were quite equal. After the analysis with GenoMAD, this program is able to detect viruses as well as other mobile genetic elements like plasmids. The number of viruses and plasmids were quite equal in all samples. And in total, they detected both about 1,000 sequences from viral viruses and plasmids. Virsorter 2 results gave us a little bit more viral sequences, and then we replicated them all over the samples to get study-specific catalog of non-redundant viral sequences called viral POTUS. And we got these non-redundant sequences, about 550. About them, 15% were proviruses, and the mean length of these viral sequences was actually quite high, 9,000 base pairs. There were also some quite long viral sequences. And actually majority of these viruses belong to the Caudophyricetes class, which is also accordant with other studies which investigate these viruses. It means that they are double-stranded DNA viruses and bacteriophages. After that, we wanted to know, are these viral fractions in fecal samples compared to extracellular vesicle samples are the same or different? We counted reads which map back to viral sequences and the total number of reads and find the ratio between them. And we normalized it to the length of contigs and to the number of total reads, but not to the level of extracellular vesicles or to the concentration of proteins in these samples. So we actually get quite big enrichment levels between fecal and vesicle probes. Vesicle fraction, at least in 3 samples, contained much higher level of viral sequences, and only 1 sample, it used to be quite the same level. We also looked specifically the different viral contigs, and we found that many contigs are enriched in extracellular vesicles fraction. A little bit smaller number were depleted in this fraction, but we found that depleted viruses were mainly proviruses, which is actually logical because they most probably come from the bacterial genomes themselves, and bacteria themselves or bacterial DNA itself is depleted in extracellular vesicles fraction compared to smaller pieces of DNA. And therefore, we can conclude that neonatal stool extracellular vesicles represent a Phage enrichment compartment, and extracellular vesicles may play important role in the development and in the modification of viruses compartment in gastrointestinal tracts, as well as bacterial compartments through viral infections. Okay, thank you.
Viruses are so much enriched in the EVs in the stool. What do you think is the reason for this? Evolutionary, or do you think there is any advantage or any reason for it?
It's hard to say. Maybe bacteriophages use these extracellular vesicles to get rid of bacteria or get out of bacteria. Maybe it's one mechanism. I don't know the real reason. It can be, we can hypothesize it.
So when you were showing that you were using different programs for determining the viral content of that, is it common in that kind of studies or not?
It's a good question. Some groups use their own programs, which they have developed themselves. In this case, they use mainly a single program, but groups who are not developing their bioinformatics software, they used to use different programs and find some summary of the results of different programs.
Okay, so do we have any more questions from the audience? If not, then thank you.
Okay, thank you.
And moving on to our next partner, who is coming from University of Helsinki, Associated Professor Mikael Nikko, who is also senior lecturer and principal investigator at veterinary biosciences at the University of Helsinki. He's leading the development interaction lab, investigating how maternal microbiota modulates the perinatal development of the intestinal immune system. And currently he is especially interested in the metabolites and EVs produced by maternal microbiota that is influencing mammalian fetus. And his presentation is combining microbiota, EVs, and immunity all together. Boris.
Thanks for the kind introduction, Christy. I will actually not talk so much about the details of specific research projects, but try to give an overall story of our joint scientific journey in OH Boost, which has been really enjoyable. I think this photo symbolizes it nicely. So here we are together, getting exposed to new things like Cryptosporidium, and look at us, happy faces. To me, this is the kind of center point of everything, like science-wise. It's ileum from a young calf, distal small intestine. It's CD45 immunostaining. So every brown dot in the figure is a leukocyte. So if you look at it, it looks like if the intestine is mainly built of leukocytes. It's a massive immune system there, but we should actually maybe call it commune system or communal system, because as you know, it's not just about aggressive defense, but it's more like controlled coexistence with the microbiota. And we try to understand how this develops and how it functions properly. And here we have, we need to understand microbiota and immunity and also extracellular vesicles. And I think we have learned quite a lot over the OS Boost project. So first of all, it prompted us to organize several very useful and interesting webinars and workshops. And for sometimes Ursula had to remind me a little bit that we should really organize these, but in the end they actually turned out really great. So we had world-class experts from Helsinki and Tartu and also elsewhere from the world to tell us about the best practices in EV research and best practices in microbiota research. And this spring we had a workshop on metaproteomics, again learning a lot. And this summer I was myself happy to participate the excellent computational immunology school here in Tartu. Everybody else was 25. You know, it's the peak of the cognitive capacity of people, very clever people from Tartu and all over the globe. I hope it also renewed my brain somewhat, but it was really full of new methods. Great course. And we are still looking forward for a couple of visits. So we will be sending a couple of people from our lab in a couple of weeks to Milan to learn about your very interesting 3D intestinal in vitro models. I, of course, didn't have a photograph of this yet because it's in the future. So I asked the leading AIs to give me a figure of 2 of our researchers traveling from Helsinki to Milan, and looks like this. So maybe no worries so far regarding the world domination and AIs taking over. At least they need to learn a little bit of more geography first. Okay, but back to the research. So we have been generating a number of research papers together, and there are even more in the pipeline. But just briefly, a couple of examples. So we have been really happy to work together with Thomas and Marina and Elizabeth and colleagues to learn more about the microbiota, intestinal microbiota of calves. And we have been providing training in the microbiota sequencing data analysis for researchers in Thomas's group. And in return, Thomas has provided us with the very important expertise in advanced statistics and early life immunity. We have been now working lately with this study. So I tell a little bit about this because I think it's really interesting also. In this study, we treated pregnant sows with antibiotics at roughly mid-gestation. It's the critical period of development for the fetal development of the piglet immune system. And then we saw what happened to the sow and piglet microbiotas and immune system. And when we look at the sows, it seems that the antibiotics interfered with the dynamics of the maternal microbiota during pregnancy. So it's supposed to change quite a lot towards the end of pregnancy, but the antibiotics kind of blunted the change. And that might have consequences because then when we look at the piglets, there were significant impacts on the microbiotas. I should mention that all this happened, so the antibiotic treatment was so early in pregnancy that the sows were pretty much back to normal by the time of birth. But despite this, there were significant impacts on the piglet microbiotas, on the piglet immune system, including the SAA phenomenon reported previously by Thomas. And this lasted well beyond weaning and separation from the dam, so it seems like a long-lasting event. And at the practical level, they grew less, significantly less. The piglets of the antibiotic-treated dams grew significantly less. And we are now trying to understand this further. Yeah, and of course EVs. So this has been a week of EVs. I want to once more thank all the organizers of the excellent excellent move symposium. I gave a talk about this study. So we have been characterizing the proteomes of extracellular vesicles from intestinal commensal bacteria. And I want to thank Subhashini for significantly improving this manuscript. I have been in exchange trying to do my best to help Janaka to improve his paper, and I was happy to see that you are still alive after my feedback. But yeah, this is pretty much where we are now. It's been a really enjoyable and useful ride, and of course I hope that this is just the beginning and we get to do many interesting things together also in the future. But for now, I just want to thank everybody.
Thank you first. Do you have any questions from the audience? Okay, while you are thinking, maybe I wanted to ask about this piglet study. So how long did you follow them, or how did you observe the changes like during the time dynamically?
Yeah, we follow them until 10 weeks of age. So in Finland, or at least in this farm, they are weaned at 3 weeks. So we sample them at 3 days of age at 3 weeks, which is just before the weaning, and then at 10 weeks. This is the practical endpoint, because in this system they then leave for the next place. So it was like 1.5 months after the weaning, the last time point. And the differences between the groups, or the impacts, were actually biggest at then. So they didn't go down, they went up, which is interesting.
This is rather a comment, actually. I think you forgot that we were immensely grateful for your co-facility for the help and also the analysis and training us with the LazyPipe software, which actually helped our researchers to dwell more into the metagenomics. So that's, I think, you missed in your part. I just want to remind it for you.
Sorry, it's a bit difficult to hear when it comes from the two.
No, I mean, I just wanted to compliment that we got your help from the Helsinki side to delve into the lot into the metagenomic part of our studies, because our researchers got the hands-on experience from your side to use the LazyPipe and all these new tools. So I should thank you for coordinating that as well.
Yeah, thank you for always very warm hospitality every time we visit Tartu.
If I can't see any more questions, then thank you.
Speaking about the hospitality.
Finishing this session with a presentation from Dr. Sharon Arcuri, who is the postdoctoral researcher at the University of Milan, working under the supervision of Professor Tiziana Previni. She holds a degree in veterinary biotechnology sciences and a PhD in veterinary sciences. Both of them came from the University of Milan. Her research primary focus on the cell differentiation and development of advanced 3D in vitro models. Currently, she's involved in projects investigating the impact of environmental pollution on reproductive processes. And we will be hearing about 3D strategies investigating the effect of milk-derived EVs.
So thank you for your introduction, and good hello to everyone. So my plan was to discuss a little bit on the Onehealth concept, but we already discussed previously with Professor Alireza, so I just move on. And I would like to discuss what was the UNIMI role in the project. So the role of UNIMI was to develop a 3D in vitro intestinal model. So as you already know, the intestine is a very complex organ, which is composed of different kind of compartments, and the main are the extracellular matrix and the connective stroma compartment, which is able to support the epithelium layer on the top. So what are the possible strategies that can we use to recreate in vitro artificial intestines? So we can use 2D culture dishes, which are very simple to use, but they do not reflect exactly the complex architecture of the in vivo tissue. And we can use synthetic scaffolds or transwell inserts, which the transwell inserts are inserts able to recreate the epithelial compartment, developing a monolayer of cells, while synthetic scaffolds are more complex, so they are able to, they permit to recreate, to develop the two main compartments of the intestine, so the stromal first and the epithelial on the top. Another strategies that can we use to recreate a more complex structures in vitro are represented by the biological scaffolds, which are very similar to the native organ and maintain the complexity of the structure. So what is decellularization? So how can we develop biological scaffolds? We can use the decellularization process, which is a protocol, a process able to eliminate the cellular component of the native organ, preserving the complex architecture of the tissue and the extracellular matrix composition. Why we use decellularization? Because we can maintain exactly the complex architecture of the native organ, maintaining the extracellular matrix as a natural biomaterial that can influence the cell fate, but also its function. So for the generation of the 3D intestinal scaffold, we collect intestine from the slaughterhouse. We process the native tissue to the decellularization process in order to obtain the intestinal biological scaffold. So as you can see here in the slide, during and at the end of the decellularization process, the tissue changed completely its color. And what does it mean? It means that the cellular components are completely removed. So what we obtained is exactly the native structure of the organ without the native cells. And as we can see here in the slides, with the histological stainings, we can compare the presence of the main component of the extracellular matrix between the native tissue and the decellularized scaffolds. And as we can see that the percentage of the amount of the main extracellular matrix components, such as collagen, elastin, and the GAGs, are completely maintained at the end of the process. If you need more, if you are interested in this work, I wrote also, I show you also our paper. So we generated the scaffold, and what we did was the challenge of the 3D intestinal models with milk-derived extracellular vesicles. So thanks to the collaboration with the EMU University, we received the purified milk-derived extracellular vesicles, and we used them in 2 different conditions. So the first group, experimental group, was the MAVS exposed cells. So what we did was to expose with milk-derived extracellular vesicles the intestinal primary stromal cells for 24 hours. And at the end of the exposure time, we seeded the cells on the top on the untreated intestinal biological scaffolds. We used 3 different MAVS concentration, 10 to 6, 10 to 8, and 10 to 10 particles per ml. All of them were treated and were used as a treatment of 24 hours. As we can see as the first results, so these are some representative images of the experimental group, so with the histological images of hematoxylindeusin and the DAPI stainings at the 3 different concentrations of MAVS. And what we can say is that MAVS used at a concentration higher than 10 to 6 particles per ml induced a statistically significant increase in stromal cell density, which are comparable to cells supplemented with FBS. So in the graph, we can see in yellow, the yellow bars, which are our stromal cells exposed to MAVS, and we can see that we have an increase in cell density, but are exactly comparable to our positive control, which is cells cultured with complete culture medium supplementing with FBS. On the other hand, the second experimental group is represented by MAVS exposed biological scaffold. So we used exactly the same concentration that I described before, and I exposed the intestinal biological scaffold with MAVS for 24 hours. And at the end of the exposure time, we had a pre-treated intestinal scaffolds. And on the top of it, untreated intestinal stromal cells were seated on the top and cultured. As a result, once again, we can see histological stainings of these groups. So the MAVS used at a concentration higher than 10 to 6 particles per ml to precondition the intestinal biological scaffolds is able to induce a statistically significant increase in the stromal cell density. So here in the graph, in the graph we can see in green the different concentration of MAVS tested. And once again, we can see that the response in cell density increased in a dose-dependent manner, and the lower MAV concentration used is statistically equal, comparable to our negative control, which is represented by cells cultured with the standard culture medium depleted from FBS. In the histological settings, we can see that cells are able to engraft, to repopulate the scaffolds, and the cell density is increased in a dose-dependent manner. So in this slide, we can compare all the experimental groups that we had. So once again, in green, we had the pre-treated scaffolds with the different concentration of MAVS. In yellow, the intestinal stromal cells exposed to the different concentration of MAVS. In blue, in dark blue, negative control. In light blue, positive control. And what we can see here is that if we pre-treated the scaffolds with MAVS, we have an increase in cell density, which is in a dose-dependent manner. So if we pre-treated our biological scaffolds with the lower concentration of MAVS, so 10 to 6 particles per ml, we have a response which is similar and comparable to the cells exposed to MAVS as well as with our negative control. If we increase the concentration of MAV, if we pre-treat it before our scaffolds, we have a statistical increase in cell density. So what does it mean? So in conclusions, we can say that we successfully generated the intestinal biological scaffolds using the decellularization process, which is able to mimic the features of the native organ. The addition of MAVs at a concentration higher than 10 to 6 particles per ml are able to induce a statistically significant increase in the stromal cell density. This effect was further boosted when MAVs were used to precondition the scaffolds and in a dose-dependent manner. So altogether, what we can say is that this data suggests the possibility to use MAVs as an in vitro enriching supplements, able to reduce or maybe replace FBS in intestinal cell cultures. I would like to thank the OH Boost, the UNIMI team, the Estonian University of Life Science, a particular thank to Madusha, who stayed with us for a 6-month period and helped us in this work. And of course, I would like to thank you, all of you, for your kind attention.
Thank you. And now it's time for questions.
Hi.
Thank you for your presentation. I was just wondering that these in vivo intestinal scaffolds that you developed, are they currently being used for some experiments, or do you predict that what kind of experiments they can be used for in comparison to in vivo? Because as you said that they are not as complex as in vivo's actual organs now.
Okay, so thank you very much for your question. So we selected, decided to use this kind of techniques because it's able to use the native organs. And with the decellularization protocol, you are able to remove the native tissue. And what does it mean? It means that this kind of native organ, decellularized organ, can use to be repopulated with different kind of cells. And of course, to test, to investigate the fact that this kind of MAVs can have on the intestinal tissue, of course, we can use different kind of strategies, for example, 2D or other types of 3D models, but the use of the biological scaffolds is able to obtain a more reliable data. So the results, I think that the results could be more near to the physiological. So what I want to say is that sometimes what happens is that when you use a model, a 2D model, for example, and you expose your cells with MAVs or whatever you want, you obtained results, okay? But sometimes these kind of results, when you translate the results in a 3D models, sometimes the results are not so near. So you have, when you are going to test something, you have to select the more reliable in vitro models which is able to guarantee the more physiological environment for your cells.
Thank you for your presentation. So I will continue this discussion you had about the reliability to the real world situation, your models. So I'm a believer anyway. I drink milk as lot as possible. However, I just would ask you the question, can I use your study to convince my vegan friends to say that maybe milk is still better than oat milk? Do you have any consideration about this kind of stuff?
Oh, well, a good question. So I didn't think about it. So, oh, wow. I have to think about this question because
Don't worry, I just teasing with you. So this is so complicated question. Then we should not talk about this. We have trouble to talk about this when we are scientists.
Mhm.
But we should talk about this when we are not scientists as a specialist with to give the message to the outside of this. I think the problem is this problem we have with all things we do in the umbrella of OHA Boost, actually, in that sense. So I actually like this very much.
thank you.
And I will use this example to convince my vegan friends that you should switch from oat milk to the real milk. Thank you.
Thank you.
When looking at your data, you're showing that the milk EVs are affecting the not the intestinal epithelial cells,
Yeah, the
but
stromal cells.
stromal cells. Is it reported that generally the whole milk has such a function, or do you know of any studies that have shown such kind of effect? And also, why you think that this is happening, or what is there any advantage to that stromal cells are getting supported and intestinal cells are not getting supported?
Okay, so thank you for the question. Okay, so in literature at the moment there are information about the effect of MAVs on the intestine, so on the intestinal epithelium, but there are very few information regarding the stroma, so the connective tissue, which I think it is very important for the maintenance of the homeostasis or for the other biological mechanisms which are responsible for the correct maintenance of the intestinal health. So I think that the connective is a particular tissue which is responsible for the protection and for the response also of the epithelial layer which is on the top. So maybe if I think about the some kind of intestinal problems, inflammations or something like that, I think that the epithelium is damaged in different ways, and the molecules goes down, and where they go in the connective tissue. So I think that if we have a connective tissue which is very robust and and the cells in it are able to respond somehow to molecules, it is a good thing.
Any more questions? Comments? If not, then thank you.
Thank you.
With that, we are ending the session and we are on time nicely. And we will be meeting for the next session 3:45. Do you have anything to add?
Yeah, if it is possible that we make a picture, all of us in front of this podium, then that would be great. Please, if you can all come, and if you stand here, we would have the whole picture before the coffee break. And yes, that would be great if we can have in the background, the boosting Onehealth.
Welcome back. So the next session is actually to talk about the individual's experience that they had during the different kind of exchange visits, exposure visits, job shadowings that we had throughout the project period. To share some of the experiences, I would like to kindly invite first Professor Arvo Wildrop onto the stage, and then Quilimust, and then Egne Kauru, and Ursula, our project assistant, please, and then Deep Bhattacharya, representing the PhD Students Exchange. So the session will go on like, I will kindly request each of you to introduce yourself, and then I will ask a couple of questions, not difficult ones actually, very simple questions, just to let the audience know what kind of experience you had and how you can relate it into the future Onehealth related activities, especially focusing the EMU. So may I ask from Arvo, from your side to start?
Kas see töötab? See töötab. Yes, it works. Yes, good afternoon. My name is Arvo Wildrop. I'm a professor of epidemiology at the Veterinary Medicine, at the Institute of Veterinary Medicine and Animal Sciences, and I'm also head of chair professor of the chair of veterinary biomedicine and food hygiene, which is directly involved in Onehealth topics in our university.
Good afternoon. Yes. My name is Quilimust, and I am head of the Joint Clinical Veterinary Laboratory at our institute.
Hello, my name is Egne Kauru, and I'm a senior veterinarian in the equine clinic of Estonian University of Life Sciences.
Good afternoon. My name is Ursula Malinska-Mark, and I'm project management assistant also in the same Institute of Veterinary Medicine and Animal Sciences.
Hello, everyone. My name is Deep, and I'm a PhD student at the same university.
Yeah, so if you see the profiles of each of these individuals, you can appreciate that we have trying to bring all the levels to gain some kind of exposure training in different aspects, specifically in their relevant fields. It could be from research, it could be from teaching, it could be from a perspective of even laboratory, like real clinical level application. So if I may ask from Arvo, how was your experience, and where were you in during your job shadowing, and if you can tell us a bit more on your overall experience and how you can relate it to the future Onehealth goals of the EMU. That would be very nice.
Yeah, my visit was I don't remember when, not long ago, in spring, I think it was in spring. And of course, my view on this visit was, or my aim for this visit was more kind of strategic, to have a more strategic view on topics of Onehealth at Milan University. So that's why I was kind of visiting various departments, food department, chemistry department, virology department, and had a good discussions there with colleagues there, and we discussed the research directions, and also I was introduced to the achievements in those departments, and then I think in that sense I had a very good time to establish new contacts course, and that's the main aim of that visit for me, of course, and collect some new ideas, of course, where our One Health research could be directed in the future, and yeah, and also maybe learn some from the goods and bads of the other universities, right? And try to learn from these and try to avoid maybe some mistakes what have been done somewhere else.
Right. If I ask from Kuli, how was your experience and where were you during your exchange? Yeah.
I had a great opportunity to visit Milan University's Lodi campus for 5 days. And actually this was a work package 3, probably know. And I tried to remember his name. It was Management of Core Facilities. It was the subtitle of our visit. So I was there together with my colleague from Weed Analysis Laboratory, Andres Velte. So our aim was actually to see different laboratories, how these laboratories manage themselves. And we spent days in very, very different labs. And also I had a chance to see university's animal clinics and animal clinics operation halls. And it was actually very, how to say, eye-opening to see other facilities and how these everyday activities are done in these laboratories. And also, of course, to meet colleagues, and then that's always very nice to conversate with people who are talking this, let's say, same language, this laboratory language. So, and yes, I think also that this main importance of this kind of visits is to have new contacts, and then you know where to ask something, where from you can get help or advice. As I'm not a researcher, I'm just a support staff, but it's still same importance for support staff also.
I think laboratory management is a very, very critical and important thing as researchers as well as in clinical perspective. So I think it was a great opportunity for you. So Egne, if I may ask you the same question.
Yes, so I had a chance to visit the Equine Clinic of Helsinki University. So I was there as a job shadow. There were different aspects I could cover, so it was really nice. Like, first of all, teaching, research, and biosecurity as well. So it was really nice to meet new people, meet the ones I already knew, and also the ones I have heard before a bit.
Thank you. So Ursula, if you may also enlighten us about your experience, that would be really helpful.
Yes, I also had the opportunity to spend 1 week in Helsinki, where I was participating in job shadowing visit together with my colleague Amita. And our aim was to shadow Topi Tanskanen as one of the project's coordinator, that he's also here today. And during this time, we had the opportunity to visit different units of the University of Helsinki structure, units named, if I remember correctly, like university services focused on project and research management, coordination, applications creation. We met different people involved in these processes, in different steps of these processes, and we had also time to reflect what kind of solutions that exist in Helsinki University we can try to implement here in the Estonian University of Life Sciences.
Thank you very much. I think among all of them, Deep had the longest stay at Milan. So can you please just enlighten us?
Yes, absolutely. So I had the wonderful opportunity of working at Professor Tiziana's lab along with Dr. Sharon as well as Dr. Georgia. And so grazie mille for that. It was really nice. Yeah, I think this was a culmination of what One Health meant for me as a young researcher, that I could go to a lab and work on pipelines and systems that they have developed and are good at, and could bring some of the EV work that we have done, and yeah, collaborative work. And it was a great opportunity, great experience. We are still working on a project and hope to publish some work soon.
Yeah, that's great to hear. Yeah, so I would like to move again back to you, Aro. Yeah, so like we all, our speakers actually mentioned, the One Health OH Boost is not just a destination, actually just a start of another journey. So we are trying to work along this journey for coming, I don't know how many years. Like the director mentioned, that we are going to now even going to have a structural unit even to boost the One Health more up to a different level. So how would you think that this kind of activity, specifically the exchanges as well as job shadowing, how would that help if you look at the future of the One Health journey in the EMU? Do you think it's something that we should continue in the future as well as, or what's your overall perspective on that?
Yeah, of course, this kind of activities, exchange, people going visit other facilities, other researchers, and to be introduced to the work more, looking really closely into that. It's really, I think it's useful, and it should be, yeah, it should be continued. And of course, in our future activities, those contacts what we have established, I think we can further develop those contacts. And then if we have similar research branches and similar directions, we always, it's good to share some experience and discuss, and that we can say, and now we know the people, we can really try to arrange the meetings and for further deeper collaboration. Yeah, absolutely, it's necessary.
That's very nice to hear that. So if I may ask from Kuli, so did you see a different level of engagement, especially in the case, since you manage the clinical laboratories, the analytical facilities here? So if you look at right now what you are doing here, and also if you compare the laboratories at Milan, so did you see a big difference in terms of research involvement? Like whether, do you see a big difference, or was it the same? Or do you think what else you can do after looking at their operation practices?
Okay, I cannot say that I saw a big difference, but it was very impressive for me that in Milan University laboratories, how to say, there are very close links in teams with research and laboratory activities.
Okay.
So very often there are people who are working at the laboratories, they are also researchers or members of research groups. And yes, really, I saw these kind of things in many laboratories. For example, in parasitology lab, there was a young researcher who was also involved, let's say, in human part of parasitology. So I also saw the very close links to other disciplines.
Okay.
They had some problems with this human echinococcosis, which is a problem in Italy, and this parasitology labs researcher who at the same time did this molecular analysis for parasitology in this institute's lab. So it was really very good to see that things can go like this. It's not very usual in our labs.
Okay, so you think that we should also move into that level of operation? Okay, that's very, very, very good to hear, actually. Yes, we have to, sometimes we have to adopt new strategies as well. Yeah. So Egni, if I ask you, since your perspective was different, you were a clinician here, and then I know when I was looking for somebody, when I asked from Felipe, so he said, yeah, we have enough people to send. You know, they love to be somewhere and learn new techniques and all that different practices. So did you gain something that, for for example, made some changes within yourself when you really reflect on your visit and experience?
Yes, thank you for the question. Like there were multiple aspects. Like for example, if you think about biosecurity, like the University Clinic in Helsinki, they built a new examination room for horses who are in the isolation unit. So it was really nice to see that's definitely separated or completely separated, and so the biosecurity level is higher this way. So the cross contamination is less likely to happen. So we are so used to the ways we do things and the opportunities we have. So if we see something different, because I had never thought about it before, to have a separate examination room. And now when I saw it, I was like, but why don't we have it? It's an excellent thing to have. Okay. So that was like the one point. The other point was that I met Associate Professor Anna Mykänen there, and I wanted to talk with her about research a bit, like clinical research and like PhD studies there. And then I met also one of the former students of ours, Nini Höglund, who was finishing her PhD studies. So it was like...
So you got inspired.
Yes, and I got inspired in a way I had never thought that I would like to do or like be really involved in research. I'm a clinician, like I want to treat animals, horses especially, but like the research behind it, like it's a different world. And like when Anna explained to me the research projects they are doing at the moment, and then like she talked about extracellular vesicles, about lipidomics, and I was like, I don't understand anything. Like every second word, yes, I can understand. And lipidomics has something to do with lipids, I assume. And so it's like, okay, but, and then when I met Nini and her research is about equine asthma, I was thinking like, if they can do it, we can also do this like similar research in Estonia, because we have a lot of asthma horses here. So it was like for me a point like to think that I might be interested in research. So let's see what the future brings. We have some thoughts about the collaboration and stuff, but let's see how everything goes.
Okay, that's very great to hear. Yeah, please go ahead. Yeah, that's what I said. Yeah. Yeah. I think it's a very, very, very rare thing that you can observe in India. Yeah, clinician is getting inspired to do. Yeah. Okay. So that's, yes, of course, definitely. So, what about you, Ursula? So, did you bring anything back with you that something that changed your day-to-day life with us? For example, I know, like you mentioned, you were sending 100 emails, 100 reminders. So other than that, what else did you really extract out of your visit over there?
My main takeaways were like two elements. We had very interesting meeting with the lady working in one of the university services that is called like this in the university structure. She was from research funding services. And the way how she presented their work, it was very inspirational, because they have this kind of system that you can have idea for any project, you can go to these people and they will guide you from the very beginning till the very end. And when I heard that at the University of Helsinki there exist people like grant coaches, I was like what? Coaching about grants? But then I understood the system, that there is the structure, that there is a process to prepare the application, to develop the idea, to help with writing the grant, to help with finding the right partners, to help with digging into the topic, what exactly you want to do. So basically it's that, I don't know if any of PhD students want to write some grant application, but doesn't have any experience in that. There are dedicated people who are guiding you through this pre-award phase, but also through past award phase. And I was very inspired not only thinking about our PhD student, but also about myself as a project assistant, that if I want to do something, and for example, I don't have big knowledge about scientific part of the topic, then I can also reach out to this kind of people, and they will help me to find others who are connected with the topic that I want to do the project about. And second takeaway was how many solutions that are implemented at the University of Helsinki is based already on AI. We had meeting with financial department, and the ladies were explaining to us how they support their work, basically applications and different programs support daily work of project managers. That, for example, I don't have to reach to financial department asking how is the situation with the budget, how much money we spend, because there is some AI-based solution that automatically is sending every month this kind of reports to project coordinators. So if they want to see the numbers or not, they still do because it's automatized. So these were like two main takeaways. The structure that is supporting people who want to do projects, who want to get money for their work. And the second thing is innovative tools that already exist and make life easier, just that we don't waste time on some small tasks.
All right. So maybe since the director is also there, maybe we can request him
Director
also
heard about all of this because just after job shadowing,
Yeah,
we had
okay.
meeting.
So yeah, it's really great that we learn certain good things from them, and then if we try to implement them for our betterment in EMU. Yes. So Deep, what was the most impactful thing that you had during your visit, and what changes that brought into you when you look at yourself reflecting your experience?
I think there were plenty, but to name a few, I would say that definitely the volume of work that they did there made me realize that, yeah, that is something that really can help one develop more confidence with what you're doing. And especially when you're entering a lab which has a certain expertise in certain things, you can learn a lot of things and also revisit your own basics and question a lot more than you usually would. Because, for example, if you're growing cells, you know this media at this much time and you're done. But there are so many intricacies that you can get into. So I think having an eye for the details and also asking why this, why not this. And yeah, I think that is very important for any researcher, but especially on a personal level for me, that was quite helpful. And yeah, I think that is
So
one of
overall,
the
you're
things.
happy and you got something to your life, not only
For sure, yeah.
Great. So is there anything else that the audience would wish to know from them before I'm going to move to the next? We can have an early closure if we don't have anything else. Yes, Thomas, please.
Yes, my personal experience with to be somewhere else is that it can define yourself better. It's the general rule what we have. We don't know who we are. To do that, we need to see something else and say, we are exactly like that, or we are not exactly like that. So my experience from this, when I have been in the abroad and stay quite a lot in Finland, was that I grow as a person because I know myself better. I was just wondering, do you have the same experience? Maybe not in this case, but in other visits, can understand you and define you as a researcher or as a coordinator or something like that. So. Yes, do others have this kind of experience in that case? Usually I realize this only retrospectively, but just wondering.
I may answer. Yeah, I got this knowledge from these laboratory visits that you very often see that we are doing things a similar way. We are doing this laboratory activities the same way as we are doing. And this is a very good confirmation that we are on the right way and we are on the same level as other labs are. But at the same time, you can also find that they are doing something differently. So that's a place to develop, I think.
My experience with this kind of stuff is that sometimes this experience is not very well suitable for our situation. So the tricky part is that they do it like that. Okay, good. And then you realize you cannot do this like that here. So what to do? And then you start to think creatively. And you can come up with solutions which are more suitable in this situation, in different environment, etc., etc. At least this is my experience.
I can give a comment about it. As after my visit in Helsinki, I heard some comments that, oh, it's different type of the university, different scale, so they have different solutions. But my approach is that the aim of this kind of activities in the projects, it's not to copy paste, but to be inspired, but by what other people do somewhere else. And exactly as you said, be creative and think, okay, maybe this small solution from there we can implement to our local conditions, and in this way make our work better. Because of course, each institution, each work type, each of us is different. We have different conditions, different possibilities, different capacity, also financial. But still, I find it attractive and interesting to go, to see, and then think, okay, so what even the smallest element, what kind of the smallest element from this experience I can take and I can implement in my daily life. And for me, for example, the possibility to have meeting with the director and to share my impressions and feelings and leave to him some food for thought, what we can change, is also this something that I can do after the visit, that maybe inspire others. Maybe he will talk with rector of the university. Let's try to do this or that. So it's always worth, because even the smallest element we can take and try to implement locally to the conditions that we have.
Yeah, so I think overall the audience have taken something, essence of what you had and what you experienced. And then obviously, so like I mentioned previously, this is not just a endpoint, so we are going to continue it and we'll be seeing more and more visits. Probably we had like 120 total visits if I look at your numbers, right, in overall last 3 years. So this is just a snapshot of what we had. I think we had many people who did many different types of exchange. I think Alrisa is going to talk about that in the next session with a few of other academics from the EMU. So I would like to thank each of you for your generous participation, because I know it's how difficult to take, especially like a person like Avo, to take 1 week out of your schedule and to help us in fulfilling the project objectives plus bringing something back. So individually, I would like to thank each of you actually for being a very active participant in the project and helping us to reach our deliverables in our terms, especially because always the EU is asking, where did you reach your deliverable? Did you finish or did you reach your objectives always? So yes, we got our ticks as well as I'm really happy to hear that you all got something out of it. So thank you very much. But we have something for each of you before you leave the stage. Yeah, this is really small token of appreciation for your active participation as well as sharing your thoughts with us today. Yeah, so we are going to close the session and move to the next session of the day. Thank you.
Yes.
Thank you very much, Suranga, and also all the panel. And I think one point we didn't mention here, I have to, both the Milan and Helsinki group, you see, you put a lot of work to arrange all of these exchanges and going and coming. I think you probably now felt that it has an impact. They are all somehow thinking, okay, it was like this, it was like this, and it was not all the energy that you have put there. It has been for a very good cause. Great. It is the last session. We are a bit earlier than the usual. I think I'm going to ask Associate Professor Kristi Huig, Professor Tiziana Brevini, Professor Heli Simoyoki, and our director, Thomas Tirarts, to come, and we would start the last part of it. It is not, yeah, please take your seats. This is not the first twinning project in this university and also in Tartu. I have been actually in Tartu University. I was one of the twinning in Estonian University of Sciences. When I was working in Sheffield University, I was somehow in the seats of Tiziana and Heli, and there were somehow at that time there was another twinning that we had there, and it was about reproduction and so going on. And I have been also, I think, Tunnel, you had also another twinning project in UT and so. And these twinning projects are part of the widening program of the European Union. And I have a feeling they are definitely, totally, the widening program has been successful in the way that it is somehow working. We can see it also in all the statistics that is coming out. Actually, as a result of the successes of the widening programs, ERA-CHAIRS, twinning, all the other ones that is coming out, Estonia, unfortunately, is going to be thrown out of the widening program because you're doing so good job with the amount of the money that is bringing to this country for research. If I'm right, the prediction is something in the last two horizons, over $500 million you have somehow brought in. It was I saw in a research communication that came from the director of ETAC that Estonian researchers have been able to do such a great job. There was even a paper some times ago, maybe 3-4 years ago, in Nature about the level of the impact of the particularly Estonian researchers, the research that they are publishing, that how high it is. And so that by all the metrics of European Union, in Estonia, we have done great. And then as a result of that, our last program, actually in last Horizon Europe, there was the question that Estonia should come out of the widening. It is quite going very well. And now what I'm hearing that even for the countries like Estonia, they are going to open a new category that we are not going to definitely be anymore in widening, but we would be in a category of transitional, but we will see how it goes. But that also brings us to our training and the work that we have done with the Onehealth. And you guys, you are actually representing for us all the different institutions. Thomas being the director of the veterinary institution. At the same time, Kristi has been also the representative from the Tartu University. Heli from the part of the Finland, the Helsinki has been our representative, and Tiziana from the Milan University. I think if you want, you can also introduce yourselves. What I think you are non-entities. Or do you like to introduce? Or yeah? Go ahead, Kristi, please. Yeah. No. Yeah.
So I'm coming from University of Tartu. and working with viruses at the Faculty of Medicine. So doing the molecular epidemiology mainly.
Thank you very much. Thomas, please.
I'm Toomas Tiirats and head of the Institute of Veterinary Medicine and Animal Sciences. Nowadays, not much dealing with research directly, but my ultimate task is create environment for successful research.
Thank you.
So I'm Heli Simojoki. I'm Vice Dean of our veterinary faculty in Helsinki, responsible for clinical environments and infrastructure. And I'm also a professor of ruminant health, so I am interested in production animal medicine, and I'm also a clinician by that count. No?
Yeah, it is working now. Good evening. I'm Tiziana Brevini from the University of Milan. I teach anatomy and embryology, molecular embryology at the Faculty of Veterinary Medicine. And yeah, I'm very fond of research, which I consider probably my main activity. I have a lab, a very active lab, mainly working with, as you probably could realize, good predictive models that tries to avoid as much as possible the use of animals for experimental research.
Thank you very much. Okay, my first question is actually towards Thomas and Christy. What aspect of the One Health work that has been supported by OH Boost do you think would be able to continue in either in Estonian University of Life Sciences or in Tartu University? Maybe Thomas, if you want to.
Probably the project has been most useful tool to create kind of a forward the message about One Health, to raise awareness of different decision makers that such processes are going on, and Tartu is here as a leading part of this movement. This is something. And I'd like to accent really that it's the right time to distribute as a message about One Health and all the different angles of this topic, because in sense of definition, One Health is very broad, very wide, and sometimes it's not so easy to recognize it. It also means kind of preparedness of future emergencies, changes. Even the climate change is directly involved, as you know. But such scenarios to make right decisions to survive, yeah, in Estonia and in a global sense, it's actually One Health approach.
Christy, what is your opinion? Do you think this program has had any effect on the relationship between EMU and UT, or generally even on UT work on the One Health?
Yes, so I think it had an impact, as we were expecting also, because I think the collaboration between the two universities, or in more precisely the labs that were collaborating, and I think this is something that will continue, because we have got familiar with each other and we know the strengths and also limitations of that, and that part is most likely to continue. And the other part is the development of the PhD program, not like maybe one separate PhD program, but integrating the different parts that will engage more One Health. Because I think, as Thomas already mentioned, the One Health concept in general is not so familiar, not only in general population, but we are talking also the higher education levels. So these are, I think, the two main things that can continue.
If we manage to do that, I think that would be amazing. Even not only at our level, you know, at the level of Estonia, if we would have one PhD program that would be integrated between medical school and veterinary. So I think worldwide we are going to be somehow very, especially integration in the sense that, and I didn't thought actually about it, and this would be a very, very amazing outcome of our program. But our partners, you were supposed to give us all the knowledge, everything. But was it always a one-way route, or did you get anything from us? What is your feelings? You can be frank. You can tell what you think as you want.
Okay. Yes, we get a lot.
Thank
like Mikael showed in his presentation. So we get a lot from there, and also from these job shadows visits, and these, they are always both sides. We ask about you, the visitors, things, and also challenge our what we are doing is are we doing right thing? So of course they are both sided. Yeah.
I think that it would be a real pity because there is such a kind of consolidated situation where people comes and go. I mean, we are used to have somebody coming from Tartu or somebody going to Helsinki, or I mean, it's become some kind of routine, let's put it like that, not in a negative sense, of course. So for sure, fundings permitting, because that's a key point, I guess. It would be very rewarding. And at this stage, I think it would be the moment when it would become fruitful, because it takes a while to start and to understand each other and to find the same scientific and behavioral language beside the language communication, which is not a problem, of course. So having said that, definitely, I think that even the idea of a PhD program like the one we have this Interreg program, for instance, European Interreg program, interregional program that gathers, for instance, Swiss, Italy, France, all the Alpine region, for instance, just putting somehow populations that have got communal traits or something in common, and the common trait would be interest for One Health in this case. So I think we have to find a recipe, a winning recipe to try and implement the system. I'm not clear what is the recipe right now, but you know, when there is a wish, there is a way, they say. So I guess.
By the way, I am not the only one that needs to ask questions, or these ladies and gentlemen are not only persons that can answer the questions. At any moment of the time, if you have any urge to put your hands up and come into the discussion, please do so. We didn't have anything from the people that were in the Zoom, in the internet. If anyone wants to, or if there is any questions or so, maybe they don't know that they can also ask, and they know, okay. And if anyone wants to come in, by all means, they can come in. But yes, Thomas, and I think, no. We need to have our collaborator, cooperator to work.
I would like to ask the question when I think 2 days ago when we had this veterinary medicine accreditation committee, and it was very panel and discussion and questions related to that. And And I noticed the strategic aspect of this kind of thing. So, as I understand, right, our strategy should be towards something we know will come. So we need to have some kind of crystal globe to say what is coming. And then we are prepared because we know what is coming. There are some problems with this in my mind. So I was just wondering about, because right now we are concentrating to the programs which are post-graduating things, etc. However, if we think as a One Health concept, if we want to widen this, then we need actually to involve from the first course of our students. So the question, I know the question is, what are the thinking about that? I know that Finns have actually thinking about this a lot, and they have started this kind of programs. What about other institutions? What do you think about these possibilities to actually incorporate this to the lower level, pre-graduate programs? And why I think the main thing is not about the knowledge, but what One Health is teach you is the same thing. It's about the skills. So what is happening so we know what to do if something unexpected is happening? We have the great example of COVID, for example. Were we ready for this kind of stuff? Probably not. But we need to spread certain skills. If something is happening, we know where to start, for example. What about your thoughts about this wider approach to spread the knowledge to the society from the beginning of the university teaching?
And yeah, Christy, Christy, please go ahead.
Start. So there is like two parts what we are doing. And so at the University of Tartu in medical faculty, we are right now designing the MD programs. And that is one consideration to start early to introduce the discipline itself. Right now, with what we are doing in sense of, so we are teaching also microbiology in medical faculty. So introducing One Health in that sense, but in broader sense, it should be in the program totally engaged. So this is what is going on at the moment, and that is one part that we are thinking of. The other part, the preparedness, are we ready or not? That is also the governmental side, because if you are looking at the targets that have sensed like what is important for the government, then looking the priority list, of course, the security, everything related to the security is at the first place. And then everything else is coming almost at the top, at the bottom. There is a health issues. And then there is a sentence, if we have time and resources, then there is preparedness for next pandemic. And I think that is something that answers how to be ready. I think it should be like coming up from the bottom if we have resources, because we all know that there is lack of resources anyway. So this is like a little bit concerning.
Yeah, thank you. Yeah, these are different parts of your question and thoughts, actually, as usual. There are a lot of angles. But yes, COVID taught us lesson that the multidisciplinarity at the crisis committee or whatever we call it is needed. And in Estonia, for example, it was very clear that the veterinary science was let out of such processes, in spite COVID considered to be animal origin. So, but the lesson learned, and it seems there are different attitude right now, a little bit. But undergraduate level, Indeed, because of this project too, we are actively working and trying to prepare different models, and most desirably Onehealth master program. And it's already something what we can make multifunctional, so-called, just to educate other professionals, and also include some certain models and the undergraduate study programs of our own students.
Okay, as Thomas mentioned, we are starting this Onehealth Master program in hopefully 27 autumn, and also actually beginning of next year we are starting Onehealth doctoral program also in University of Helsinki. And one thing I want to mention, I think that because we have talking about Onehealth in Helsinki about 10 years ago, or started to talk about it, I think nowadays nearly every teacher is always thinking through Onehealth, and it's affecting our teaching. So that's the way how these undergraduate students hear it from our voices. And then we have these shared professors quite a lot, and it helps with this multidisciplinarity because they are connecting faculties. We have several professors with medicine, Faculty of Medicine, and also two with the Faculty of Agriculture. So these are very good thing to combine, like you two. So I prefer they are very important.
Okay, so if we talk at the university level, the Faculty of Medicine has a doctorate in Onehealth and is dedicated to it, and is a 3-year course, but that's of course somebody for specialized people, so it's nothing to do with the kind of dissemination or making people more aware of what's going on. There are different levels, I think, in our country, and probably, as you know, COVID hit very hard Italy from the very beginning, and that made very clear what zoonosis was to everyone. And so this somehow started a campaign of attention towards the possibility of interactions among different kingdoms as something much more real than what you read in books, okay? It could be just outside your door. And the other aspect, so there is a lot of campaign addressed to undergraduate school students and high school students. So in general, we have what we call school ambassadoring activity, and there are specialists, or maybe even less specialists, that visit school and create initiatives on a certain periods of the year, and allow youngsters to enter the laboratories and deal with the very simplest aspect related to One Health, prevention, public health, and this area. Well, the other problem that we are facing is immigration. As you know, Italy is one of the countries where really huge waves of immigration arrives. And this brings an increased attention to diseases that basically did not exist anymore in our country. And due to this cultural changes, which is part of One Health as well. I mean, it is really big waves of movement that are taking place in the world and create new realities that we have to face. And this is another point that where there is a lot of attention and attempt to create awareness around, for instance, diseases that were somehow forgotten or, you know, some kind of solved, let's put it like that, with what we know in terms of progress in medicine and all these aspects. And the third very strong aspect in our country is sustainability. This is again something that is attracting the attention of people and where we address a lot of work, because people want to know what they eat, the quality of what they eat, how do you grow this animal, what are you doing with this animal, what these plants are done with, are they transgenics? I mean, you know, all these sort of things. And these are somehow all things that are going the big pot, at least in my way to look at the. So there is for sure a lot of attempt from the scientists and even from journalists and other area involved in this that are trying to increase awareness and knowledge.
Thank you.
What is also missing, and I think Helsinki University have been addressing that maybe more, or by the trainings at least I have heard, is the social scientist part of this communication part, but also doing science to understand the communication. And this is also part of the preparedness for next pandemic or whatever crisis we are facing.
Then I make a conclusion answering what Thomas said. And Thomas himself, in his slides, he showed nearly 20 years ago, he was doing OHA Boost. He was doing One Health Boost. But fortunately, that initiations and so has been continued here in Tartu. We have had also my own era chair that was related to the One Health, boosting again the capacity that we have here for One Health. And we have also now still running this twinning program for One Health boosting. Then when it comes to the accreditation, that I'm very glad if they have asked and brought it here, because I was not here, I was in the meeting. But if that has been one of the aspects, we already have prepared even a part of the curriculum as an optional curriculum Suranga has put there. And so interestingly, when we did go actually and asked to have this on the curriculum, one of the issues that we got was curriculum is too packed. It is not possible to add anything anymore to it. Just leave it there. But now after this comment, if it has been as one of the comments, I think we can go now back and say, hello, we have prepared this, bring it in and use it. But the good point is we have built the capacity. We are prepared. Even before they say it, we have had it, we have the module made, and it is lovely to hear that you as the professor here, you are thinking, I don't want to anymore think about postgraduate, I want to think about undergraduate. I want to see how can we bring it into the undergraduate curriculum. And hopefully even as the others mentioned, that in near future, even the clinic, in every aspect of the teaching of the veterinary medicine, we are going to look at it exactly from the point zero to bring it in. And the only thing I can say is this time we have predicted what the crystal would somehow be there, and we are now prepared for it, and we can just very, very easily, very fast, if it comes as a point, to implement it in our program. If you are happy, then Todd.
I can comment a little the same thing you said. It's it is related to the idea that we are ready, and this, as I understand, we're right now in the university doing strategical things, and we here in Institute, Thomas can confirm, we are not very pleased what the university level is thinking about that. So one way we need to emphasize, actually, I think, Thomas, you agree, is the One Health approach. Because the university itself, if they discuss with government, is also have brought this issue up. So the One Health approach in that sense. And there is another thing. I also think, because last 15 years, I have spent every year to read hundreds of veterinary student motivation letters. So, and what I have seen, that everything which is happening in the society will reflect from these motivation letters. And in one point, I started to see this kind of, this year first, I saw the One Health mentioned in the motivation letter, although I think it's because of use of AI, but anyway, I'm glad that this is there. So this kind of thing, which is related to the importance of animal health. So even the student candidates already are prepared, actually, to take these measures, because they know about the importance of society, this kind of approach to the society, to the environmental issues, etc., etc. So I think we need to try to do this, start this from beginning. Like I started 25 years ago, didn't know nothing about EVs, didn't know nothing about One Health. I realized it's One Health only 10 years later when I was introduced to the term. So that was my idea.
This is the second time today in the afternoon I have been moved. First, by hearing a clinician who wants to become researcher. Now hearing that the students, member of the public, writing motivation letter and is mentioning One Health in his motivation letter to come to the vet school. I think all of these are good signs. We are somehow progressing. If any of these guys would have something like this, they would be very, very jubilant, very happy. Then I think very well done to all of us for the, and actually this is really a work that we are all doing together. We are from every aspect that you, from the cafe scientifics to everything else, so that we are doing. Tunnel with all his work that he's doing on antimicrobial resistance, everyone, that all of us that we do the work, we are someone doing together. And on that aspect, we just heard from Thomas that we had the first reading of establishing of the One Health Center in the Estonian University of Life Sciences. And you don't know how much joy that gives to me to say that we had this first reading for the Senate. And from what I have heard, it has been taken very positively. If another time, as One Health is a very large and general topic, and with the resources that we have, till the time that, as Christy said, till the government realized that it needs to be on the top of the agenda, still we have a long way to go. By the way, I have to say that, sorry that I have to mention this always, Estonian research centers, Estonian excellence centers that we applied for in the between, I don't know, 100, 120, 150, they selected us between the 13 successful ones to go to the second stage. And then in the second stage, 10 were selected and 3 were not selected. And one of those that was not selected was ours on the One Health Institute Excellence Center. And one of the questions in the panel they asked me was, what is the economical benefit of having such a center for Estonia? I was just thinking, what should I answer to this? You know, this is what, and it was one of the advisors of the government that was asking me. And I was just, but still it seems, yeah, there is a very big lag between the way that we think and the way the government of Republic thinking. But if you want to somehow think in future, the future that would be the next 5 years, which aspects of One Health would be more sustainable and profitable for us, that we emphasize on it and try to, because we cannot cover everything. If that were the case, what do you want to would be your side? Does anyone has any idea where we have, especially in the new Onehealth Institute, that center that we are going to build in, hopefully in the Estonian University of Life Sciences?
Okay, yes, just very first thoughts about this indeed. As already mentioned, the preparedness, the geopolitical situations doesn't let us any other option. And there are also many, many topics actually to discover, not only related to this friendly neighbor we have, and then resilience because of climate change and everything. So this is also something very broad and different issues to discover.
Any other thought? Any other?
Probably I have to say education, because I think it's like we can't be out of it. And it's most, like in the world, the studies have shown that this approach is most sustainable and it's quite basically understandable. The other part about the other part also, I think the interaction between industry and public sector would be like boost sustainability. However, in Estonia, as all you know, the industry is not strong. In Europe, we can say, but in Estonia, probably it's not like something that is money-wise there. However, I have to say that it's not only sustainability by like reading money only, but also advocating and understanding the importance of that. The One Health not only, but studies have shown that One Health research right now is still mostly human-centered. And I think because of that, like we are trying to understand the sustainability, how it is beneficial for human, the crisis, the economical wise. But I think the broader view should be looked at, how it is still beneficial for humans, but how is it like combinedly beneficial, not like directly, but maybe indirectly also. That's
Before I ask the rest of the panel, I just want to say, Christy, we have made even a member of industry to voluntarily come here. I didn't expect that Ken to be here, but voluntarily be here, sitting there and listen. And he has chosen his Friday afternoon rather than being, I don't know, 100 different places that he can be, to come and say. Then it looks that we are even moving that part of the equation. It is somehow we are getting industry also involved, considering the things. Any other word of wisdom, and I fully agree with both of you. Preparedness, bioresilience, education, industry. What else should we focus in this new center that we are trying to develop in our university? And yeah, when I say our university, we are very integrated. It's not even only the Estonian University of Life Sciences, it is a combination also with Tartu University, and we are really closely working together. What else do you think? Don't be shy. Put your, and that would be helpful because, yeah, topic, please.
Yes, hello. So kind of continuing on the topic of education and then this, what you could be doing, if I understood it right, we were mainly talking about the education of the people who are in the field, like veterinarians, medics, and other closely related professionals. But then there's the question of educating those who are doing something completely different. I think nowadays most universities have some kind of obligatory sustainability course, at least in Helsinki and many of our partners. Whatever you study, you have to do a couple of ECTS points in sustainability science. And like Alireza, you mentioned, you were asked by the politicians that what is the economic benefit. which reflects that the lack of understanding that how the choices we make in our economic production, international trade and international movement, healthcare, food production, whatever, how fundamentally they affect, and they are actually the things that create the One Health problems. Then when we are handling a pandemic, we are already dealing with the like endpoint symptom. But could your One Health Center provide this kind of training also, a tiny slice of it, to people outside the core health fields, and then actually dealing with those students could actually bring a lot of input for ourselves, like in the other way around, to see how they react, how they see those things.
Thank you. That is exactly Topia. We need to change. We need to somehow think about it from these different aspects and to be able to bring. Yes, please, Thomas.
I think we underestimate the social, psychological aspect still, and we underestimate this also in veterinary medicine. I'll give you an example. We did one study which was related to the longevity of cows. Okay? And this longevity of cows in the farms is related to the welfare issues, many other issues, economical issues, etc., etc. So during this, we also included the evaluation of workers who work with cows. Okay? And we measure also infections, poison, this kind of stuff. And in the end, we work out that the results were, for me, eye-opening. So if we look at the outcome on longevity as a whole, then approximately 30% of the variability in this was explained by the workers' personal traits, their psychology, things. So what it actually means, we have concentrated everything right now in our research to look at the infections, management buildings, this kind of stuff, economical stuff. And then we realize that quite a lot of things we don't even see what can affect, what can be related to this kind of issue. So I think the social aspect, psychological aspect, is something which we lack in veterinary medicine, at least. We need to really do the good cooperation with social scientists. That's my take. The other take is related to my experience with my children. My son was in the Tartu University learning in the bachelor degree program of geography, and right now my daughter is in the biology bachelor degree. And I need to say that this kind of overall, more like One Health approach is better in geography than in biology, which is strange a little bit. But in geography, they deal very much about the same thing, human geography, which is related to that, environmental geography, everything like that. But unfortunately, in biology, there is lack of this overall view here. Because of that, I think this kind of approach, especially in this bioscience fields, should be better incorporated somehow. That's my take.
Thinking that we are reaching actually to the time limit that we had, or so we were supposed to finish this around 5:15, 17:15. If there is any other contribution or any other points that you want to somehow bring in, please do. Otherwise, I will somehow try to go to close the session and relieve you for the next afternoon of Friday to have a bit of time for yourself. Is there any other thing, any burning thing that? Okay. Excellent, Gomi. I'm so glad, Tanel, you decided.
Yeah, so in the end, I just want to advertise Onehealth event. So come back here on November 19th, where we have International Antibiotic Resistance Containment Day, and we have the awareness event here, starting at I think 11 o'clock. And there we have, I think, here it's always like difficult to have like equal contribution and representation from different sides. So here you hear that the partners are from veterinary departments, and only Kristi here is from human medicine. There I think we have the veterinary and human really 50-50, and we have quite strong contribution from environmental health. So we made it more balanced. So come and see how it goes.
Definitely. When it would be on November 19 November, definitely. And we can actually also send it in our own. Great. Then if you can assist it here. I can close the session and also have the thing for you. If there is nothing, one point I think I can make here. We are going to close everything together. Oh, yeah. Oh. Yeah, okay. Yeah. Please, we are going to have your presence. Okay, just to close the whole program. I think the feeling I am getting is we have the structure. We have reached a situation that we have the One Health as a common attractive point that is somehow putting us together, and we are somehow happy that we have the how can I say, the statue is there. Still, it hasn't reached to the state of David of Michelangelo, but just as they didn't build Rome in one night, I don't think this would be something that we think that in just by one program or so, it would be somehow possible to have it. Probably you're trying to find yourself in this composite picture. Yeah, try to see, this is it. But it is immense, it gives me immense pleasure to stand here and just think that, okay, where we were probably maybe 8 years ago, something like that. And now I know 20 years ago you started, but as it is going, and so I think especially for somebody like you, you should be very, very proud and very, very happy that something that from 20 years ago you have started, it is still getting continuously going forward. Many other fields in the science we have seen that have gone up and have crashed. They haven't gone, but this is still going on, and we are still thinking that we can even improve it, making better till it becomes the David. There is, as you heard, a number of initiatives at the moment going on here in Tartu, and another example was also the antibiotic day that is going to be in November. I have a positive view that we are going to prevail. At the same time, I have a positive view. Government is starting to hear our voices, and they are also starting, especially because of the geopolitical situation that we are in it. They are starting to take this message very seriously, because if they are not doing that, the next time, with the next COVID or the next disease that would come, we would have much problems. And especially for a country with the size of Estonia and with the population of Estonia, it is a matter of life and death. we don't have the situation like Ukraine with, I don't know, 40 million population and being able to sustain if there is would be. We are much, much smaller. In that sense, we have to be much more alert. And we are showing it also that we are very active and we are trying to somehow keep ourselves prepared for the future unpredictable issues that may come up. On that account, I want to also emphasize that OH Boost is still not finished. It would be finishing end of the year. Yeah, Topi is here. But this is the nice point of the having the end meeting somehow, end conference now, that we know if there is anything that we need to fix it, we can fix it. This is maybe after quite a lot of experiences of doing project management in the European level, finding out that. Great. I wanted to emphasize on that. In addition to that, I would like to, say that it has been a wonderful journey to be with all of you. And this work, we didn't do it just for the project itself. It is something that we really believe in it. And we think about it as an approach that we have to take and go on. Hence, that is something that we are somewhat trying to further going. And let's hope for the best and how we can go further. Thank you for your time and thank you for being here. Thank you. I forget always something, and this is the one I forget. You are here not because of me. You are here because of this lady that is sitting there and all the other guys that have been behind the scene working very hard. But can we have a very huge round of applause for that lady sitting there? And shall I give her on my own behalf? Shall I give her one of these black bags? Oh no, you can have two. Yeah. Thank you very much, Ursula. Great. Thank you.